6 o'clock, so we'll go ahead and get started. So welcome to the September meeting of the DNA Interest Group, Iowa City. We do meet here monthly, and so the fourth Tuesday of each month at 6 o'clock is our normally scheduled meeting. And I see a few new faces here. We have various topics in relation to mostly the commercial DNA testing marketplace, and so those individuals that have tested with 23andMe or Ancestry or other options, we have topics each month that are pertinent to those types of tests and what you can make out of them. Tonight we have a talk on the complex nature of traits, which would be relevant to any of these testing platforms that predict traits from your genetic data. Next month, and so October, the scheduled topic is to do tips on getting more from your Ancestry DNA test, and so we'll focus on the Ancestry platform and sort of the hidden features that the casual user may not be aware of that are on the Ancestry DNA platform and what you can use those for. And so that'll be the fourth Tuesday of October, which I think is the 23rd, maybe, something like that. Next month. And for that meeting, we will be meeting in the Digital Media Lab on the second floor of the Iowa City Public Library instead of here.
On the second floor?
Yep, on the second floor. And in that room, we have access to computers. You can bring computers, and so the subject of that may be appropriate that if you have an Ancestry DNA test, that you either bring your access credentials and use one of the computers there, or bring your own device to access your Ancestry DNA account as we go through these particular hidden features of the platform. So that's October. So tonight, what we're going to have is a talk about the complex nature of traits. Leo Brugman is going to give this presentation. He's a fourth-year MD PhD student at the University of Iowa, and he's also worked this summer as a fellow of the Personal Genome Learning Center. And so Personal Genome Learning Center is a group of students that I work with at the University of Iowa, and Leo and another graduate student worked on this summer to prepare this talk, and then if you didn't make it, the August talk, which was about traits in relation to DNA. And so I'm going to turn it over to Leo, who's going to talk tonight.
Okay, so is the mic on? Can you all hear me in back? Okay, awesome. So yeah, my name's Leo. As Bryant said, I'm a fourth year, and I'm currently working on my PhD in genetics, so fitting group to be talking to. And I'm in Dr. Jake Michelson's lab in the Department of Psychiatry. And my research is mainly focused on psychiatric genetics with a special emphasis on autism. And so I'd be happy to talk about those things as well in more detail. But tonight we're going to be talking about sort of complex trait genetics, which is partially overlapping with that psychiatric genetics. Okay. So to get everybody up to speed, we're going to do kind of like a lightning tour on the background in genetics. And so here you can see sort of a diagram of a cell in the upper right. And in the nucleus, we all have 23 pairs of chromosomes. And in total, if you kind of unravel them, as done in that image there, you would see that across all the chromosomes, there's about 3 bases in sum. So the individual sites, or bases, or nucleotides, are what's kind of shown by those T's, A's, C's, G's. And you can see that in this particular region of the unraveled chromosome, there's a gene which is being coded for. So here we're saying that's a gene. So it turns out a small portion of your DNA codes for genes, and that's the main functional unit of our genome. So genes create proteins, which are down here, and those are kind of the movers and shakers, right? So that's your machinery within a cell. Could be responsible for cleaning things up within the cell. Maybe a cell would like to move around within its tissue. So any sort of function is usually mediated through proteins. So genes code for proteins, and it turns out there's around 22,000 different genes, depending on how you define it exactly. And we care especially about these parts of the genome because differences and variation within genes has downstream effects on our traits, and that's what we're here talking about tonight. So an important thing, and also the topic of tonight, is variation within individuals. And so if you were to unravel two people's chromosomes and kind of compare them base by base, you would see on average that overall they're strikingly similar, but 1 out of every 1,350 bases, there's actually a genetic difference between two individuals on average. So part of that variation between individuals is what's called SNPs. And so being in this group, if you've been here before, you've probably heard a lot of talk about SNPs. That stands for single nucleotide polymorphism. And what it is it's a certain location in the genome, so highlighted here in blue, in which there's variation in the population. And so in this case, you either have an A at this site or you have a G. And if you were to look across the whole genome, it turns out that there's around 300 million SNPs, or 1 every 300 locations in the genome. So there's quite a lot of places in the genome where people can differ. So, okay, there are all these sites that are different between people. Why do we care? How do we use it? So the genome is huge, as I mentioned earlier, and it turns out if you were to actually print it out, kind of shown in this photograph, that it would be this large bookcase and filled with thousands and thousands of pages and many bases. And so this is kind of like a cool little display at the Broad Institute, so you can get a size of the magnitude of the genome. And in genetics, we're really interested in traits or diseases, and we would like to find parts of this genome which are influencing some trait or a disease. And so let's just visualize that by being this red lightning strike. So this red lightning strike is somewhere in the genome, and it's contributing to some trait. One way you could find that this page or half page is influencing a trait would be to go through and kind of read every line of every book between a bunch of people with and without a disease. But we could also do it in a much faster way by using SNPs. So I've told you that there's about 1 SNP every 300 bases. So this page of this book from the library is about 3,000 bases long, so you'd expect to see roughly 10 SNPs. And because these SNPs are so close to, I think this is where, the SNPs are so close to this disease-causing loci, you can actually just look at the SNP. Since they're on the same page, they'll tend to be inherited together. They're said to be linked. And so we can study what's going on at this site by actually just looking at the lines around it. Okay, so this is an example of how you might actually try to associate a given SNP with a trait. So we're going to be talking about height a lot tonight. So let's have height on our y-axis here. So increasing, you get taller as you go up. And each dot in this graph will represent a person. And so in this case, you can see we're looking at a single SNP. And on the left, we have all the people who have an A and an A. Those are their variants. In the middle, we have people who have 1A and 1G. And on the right, we have people who have 2 Gs, right? And looking at the graph on the left, it's pretty clear that there is a relationship between how many Gs you have and the height that you are. And so from this, we would say that this SNP, it seems to be influencing height, right? And if it had a negative slope, then you would you'd also say it's influencing it, but in the opposite direction, so it's decreasing height. On the right would be a case in which we see, so no correlation, no effect of this SNP on height. So as you can see, it's a relatively straight line. There's no slope. Slope is equal to 0, so we think that this SNP is not contributing to height. So that was kind of like a continuous scale, right? A lot of times we're interested in a disease, which is kind of a yes-no thing. So in that case, you can have the same sort of columns here, right? So your genotypes, each dot still represents a person. But now you're going to kind of count the number of people who either have disease or no disease and these various different like numbers of variants. So just looking at this, it's pretty clear. You probably notice this huge grouping of people who have 2 G variants and who also have the disease. And so in genome-wide association studies, we often want to kind of quantify. So how associated is this G variant with a given disease? And so to do that, we're going to take a quick step through the numbers and establish this term, which is an odds ratio. Odds ratios are very central to understanding things that you might get back from 23andMe, and so I think it's helpful to see it derived at least once. We use this chart here, and we can make this table on the top right. So to do that, you see we have our number of A variants and our number of G variants. Okay, so let's calculate what's in that top right box. So that's in our disease group, how many G variants do we have? So we reference our sample, and we see that in our disease group, there's 8 individuals who have this double G. So they have 2 copies of G. So you would say that between them, there are 16 copies of G. Still in the disease group, there are 4 people who have 1 copy of A and 1 copy of G. So one copy of G multiplied by the 4 people, that gives you 4. Add that to the 16 here, and you get that upper right corner. Right? So you can see that it's basically counting the number of G variants versus the number of A variants, broken out by disease or no disease. So then we can go ahead and quantify this effect and say how much more prevalent is our G variant in the disease group, and that will be an odds ratio. And so to do that, we divide, in our numerator, we take this 20, so 20 Gs divided by the number of As in that group, so 20 divided by 6. That's your ratio of G to A in your disease group. And divide that by the same ratio, but now in your non-disease group. So when you do that, you would get an odds ratio, in this case, of 11.1. So when you look at your 23andMe results, and they're referencing, if you have ApoE4, it has an odds ratio of this and that. This is how they are calculating those things. So what you might notice by looking at the equation for how you calculate odds ratios is if there was no difference between your 2 groups, then you would have an odds ratio of 1. And if there was sort of the opposite enrichment, so if Gs were less prevalent in your disease group, then you would have an odds ratio below 1. So this odds ratio is really kind of quantifying the strength of that variant's influence in a disease. So to tell you kind of a back of the envelope sort of thing that you can do with odds ratio and make it more than just a number, this holds for most cases, but you could take the prevalence of a disease. So let's say, myocardial infarction is at 2% of the population. And then let's say we have this SNP, which has an odds ratio of 11, and you have that SNP. A quick way of getting an estimate on how influential it would be to multiply that prevalence of the disease by your odds ratio, and then this would get you close to the prevalence of the disease in a group where they have that SNP. That's usually true. Okay, so now we've talked about what SNPs are and how we can kind of quantify their effect on a trait through odds ratios or looking at the slope of how they affect a trait. So all a GWAS is, which if you've heard that term before, it stands for a genome-wide association study is, it means that you just go through and you do that on a ton of different SNPs throughout your genome. So before we were showing the effect of one SNP, imagine doing that 800,000 times, and you report those odds ratios for each one, and that's a genome-wide association study. So once the technology became available to do this, we entered the GWAS era, and we started GWASing everything. So as kind of a quick example of this, if you go to PubMed, which is a collection of articles in the sciences and search for genome-wide association study, require that it be in the title, because that's typically how those articles will be titled, you'll get around 2,500 hits. And so those would mostly be made up of individual genome-wide association studies. So there's really been this explosion of GWAS, and they've probably measured any trait that you can think of. So these are some kind of big examples that have come out of GWAS, both ones that already exist and things that are upcoming. So UK Biobank, which you might have heard about, came out a year or two ago, and they had 500,000 people, and they studied so many traits in these people, about 4,200 traits. So could you imagine filling out that many questionnaires? So these people told them everything about anything, any trait they could measure, and health records. And so it's this really comprehensive resource on the genetics of most traits. All of Us is aiming to have 1 million people, and their emphasis, it's a US study, and it's on trying to target more diverse populations. Last month, we talked a lot about how there's been a bias towards like Western European populations, and so this study is trying to get a more diverse view. And then 23andMe is up. I think they're over 5 million people now, and they have released a number of genome-wide association studies as well. Okay, so we're now going to talk a little bit about some results. These weren't necessarily all found through GWAS. Many of them predate that, but they are some of the stronger effects that these SNPs can have in influencing a disease or a trait. So one of the big ones that we've probably all heard about is for breast cancer. And so the BRCA1 and BRCA2 variants, if you have those, can really increase your risk of getting breast cancer. So the odds ratio for those is around 3.6 to 6.9, just to give you an idea of the magnitude that we're talking about earlier. And so if you look in the general population, it's in about 12% of people, this breast cancer. This is in women. And if you have either of these variants, you can see that the prevalence of breast cancer goes up about 4-fold. So this is one of the strongest known effects, and it's reported by 23andMe and many of those companies. And down below is kind of a graphical representation of the number of people that you would expect to see affected based on their variant status. Another one, which you've probably also heard about and had tested yourself if you have 23andMe, is the ApoE4 variant for Alzheimer's disease. So the odds ratio here, if you have 1 copy, is 3. 2 copies is 14.5. So really, these are high magnitude, very big odds ratios. And it's pretty common. So 10 to 15% of the population has this ApoE4 variant. And this graph here is basically showing you the age of onset differences between groups with and without the ApoE4 variant. And so you can see in the red and blue curves that they're left shifted and that they're high, which means that they have an earlier onset and there's a high risk of getting this disease. And that's for cases where you have both of the ApoE4 variants. And then there's kind of a dosing effect. So if you have just one, then you see it's a little bit more right-shifted. It comes down a bit. And then in the case where you have what most of the population has, this ApoE3 in both, then that becomes even lower and even more right-shifted. So this is another case of a variant which, if you have it, has a pretty strong effect. For the last example of a variant with a very strong effect is my favorite example. So this is for Warfarin. So this is a case where it's really impacted medical care. So Warfarin is an anticoagulant. And so typically if somebody has a clot or a stroke, you would want to put them on Warfarin. And it's very important that you get the dosing of Warfarin correct the first time, because if you give them too much Warfarin, so they're too anticoagulated, they're at risk for a bleed. If you give them too little, then they could still have the same problem, develop another clot, and that would be pretty bad for your patient. So they studied Warfarin, and they said, well, why can't we just give everyone the same dose? And the answer turned out to be genetic. So this is a drug label here, straight like from the FDA that would be printed and provided with Warfarin. And it turns out that there's two genes, so VCORC1 and CYP2C9, which have variants in them which really control how quickly you turn over warfarin. So the effects can be pretty dramatic, where in the top left of the graph, if you have this genotype, you might need 7 milligrams of warfarin, whereas in the bottom right, you might need only 0.5 milligrams of warfarin per dose. So these are just examples of medical cases where SNPs are already kind of informing care. Okay, so I've just told you kind of the great things that variants have sort of informed us about in medicine. But there's been a much more typical result when we do these genome-wide association studies. And so to kind of show you what that would be like, I'm going to review something in my field with you all, which is a GWAS of schizophrenia. And this will be kind of like an archetype for what thousands of those GWAS studies have sort of found. So this is a paper from 2014. It was the largest GWAS at the time. They had 37,000 people with schizophrenia and then around 120,000 controls. It was a good target to study with GWAS because schizophrenia is 80% genetic, with the remaining 20% being environmental. So if you go in with this many people, you should expect to find some really robust things, and who knows, maybe we'll have a new way of diagnosing schizophrenia. So the results that they got come out in this form. So the odds ratio that we computed before is basically what GWAS is going to give to you, right? So you put in hundreds and thousands of people, and basically what you get out is a table. And so I know this is hard to read, so I'll just tell you kind of what's going on. We have the name of the SNP, we have something about where it is in the genome, and then we have essentially the odds ratio. So if you were to look at the odds ratio, you would see that they're really small. And if you can't read that, all those are, it's 1.07. So remember, from breast cancer, we had 3.9, or we had up to 8. In ApoE4, we had 14. This is 1.07. So very close to not having effect, which would be 1. So let's see. We had that shorthand notation for what we can do with odds ratios to sort of see the disease prevalence, whether or not you have a SNP. So in this case, let's do the same thing with schizophrenia. So the rate of schizophrenia is 0.25% in the population, or 0.64% of the population has schizophrenia. If you take the strongest odds ratio from that table, it was 1.32. So if you were to have that SNP, then now your rate of schizophrenia becomes 0.33 or 0.84%. So if you were to tell somebody that your risk went from 0.25% to 0.33%, they might say, I don't care. And this is an effect where you're not going to go into a psychiatric clinic and say, we need to know this person's genotype at this location because that'll help our diagnosis. There's much more pertinent things that can be looked at. So this was a very typical result from GWAS, was that they identified a lot of different variants that they can say with confidence are contributing to a trait, but the effect is so minuscule that it's really not clinically actionable. Okay, so that kind of segues us into the topic of conversation, which is sort of the nature of complex traits and complex diseases, which schizophrenia is. So this is kind of a classic genetics photograph to show. And what you can see is people lined up by height, right? So you get taller if you go this way, and they're stacked if they have the same exact height. And there are a few interesting things to notice. So this shape is bimodal, right? So you can see a little dip here. And that's because you have males and females mixed together, and they kind of have different heights on average, and so you get this dip in the middle. And you also see that there's great variation, right? People can be of very different heights. And this all exists among healthy people. Okay, so it turns out that when you're talking about traits, there's a lot of different types. And they vary in how complex they are. You have very simple things, and then you have things which we don't understand too much at this point. So on the very left would be examples of these simple traits. And so this would mean that maybe you just have one gene contributing to a trait. Examples of this would be things like albinism, wet, dry earwax, which 23andMe reports, and same with lactase persistence. So these are examples where it's very simple to figure out the genetics of these things. And we can just test you and reasonably predict what you are. If we move more towards the middle, and now it's maybe 2, 3, 4 genes contributing to a trait, you start getting things like eye color and hair color. So at this point, a lot of these are pretty worked out. We know what contributes to eye color and hair color. And yeah, so these might be like moderate complexity traits. If you move towards the right, this only shows about 6 genes contributing to a trait. You can see you have this wide variation, and that looks like the kind of the height diagram that we showed earlier, right? So you have a lot of different heights when you have multiple genes. So now you start getting into complex disorders. So these would be things like height, weight, intelligence. I put some diseases on here, schizophrenia, cancer, but there's a huge list of things which we would consider complex. And the scale isn't totally accurate here. The number of genes contributing to a trait, I have them lined up with 6, but these traits probably all have at least tens, probably hundreds, and sometimes thousands of genes which people think contribute. Okay, so we're going to talk sort of as an example about height. It's the most classical complex trait. It probably is the most studied trait. People are pretty comfortable telling you what their height is, and it's easy to collect that data, so we know a lot about it. So it turns out height is around 60 to 80% genetic, so with the rest of that being determined by environment. How much did you starve at any points? Did you have enough calcium? Things of that nature. And it turns out the bulk of that is from SNPs. So this is 50% overall, not 50% of this number. So these SNPs, these single nucleotide polymorphisms, your genotype or what variants you have are really contributing a big part to what your height is. So I've just included the table for the schizophrenia again, but this would be the same sort of table that you would get for height. And so it turns out if you look for height at the kind of results of GWAS that you see sort of the same thing as schizophrenia, that the odds ratios of your top variants are still very low. And if you were to just look at somebody's genotype at these significant loci or these significant variants individually, it wouldn't really inform you what that person's true height is. Okay, so this is kind of where the innovation comes in. So people said, we've done thousands of GWAS, and we're not finding these, like, we're out of the BRCA ones, so how can we use what we're finding to come up with some sort of reasonable way to use this data? So somebody had this idea, and they said, well, let's just look at the effects of multiple SNPs together, right? So it seems obvious enough. It took quite a while to come around. So to do this, you can kind of calculate what is referred to as a polygenic risk score. And this is, I'm going to show you the math behind it. It's kind of an oversimplification, but I think it gets the idea across. And so let's say I'm an individual with these 5 SNPs, which contribute to height or some trait. All you need to do to calculate a polygenic risk score, more or less, is to add up the odds ratios of those SNPs that you do have. And so if I had these SNPs here, 1, 2, 3, 4, 5, I would just add up the odds ratios that they have listed here, and I would have a polygenic risk score of 5.2, let's say. Okay, so if you do this in thousands of people, and let's say in this case we're studying schizophrenia again. Okay, so our controls are just healthy, regular people without neuropsychiatric diseases, and then our cases here would be schizophrenia. So you could measure the polygenic risk scores in people with schizophrenia and people without schizophrenia and see if this score is able to separate the two groups. So that's what this kind of a diagram is showing, where we have 5 different loci, so 5 different SNPs in this case, and each column here vertically would be a person. And then the color denotes whether or not at that locus, if they got a high-risk variant, one that's predisposing them to schizophrenia, or a low-risk one that doesn't have an effect on schizophrenia. And so if you kind of organize people by how many high-risk variants they have, you can see over on the right, these people all have 5 out of 5 of the high-risk variants. And you then kind of visualize these polygenic risk scores between cases and controls. You can see that you can sort of tease apart these two groups based on their kind of the additive effect of these variants that they have. And so with this, if you're a person who's, let's say, at this sort of column denoted by this green arrow, you could say, here's my genotype, what's my risk of having this disease? And so the way you can do that is you sort of draw a red line here, and that's separating, everybody to the right side of that has at least your risk or more. And you could just count the number of cases versus the number of controls, and that would give you an idea of your risk for that disease. So there are some caveats here with the math that would have to go into this, but in a simplistic way, you could say there's an 80% chance that we think you're a case or you have schizophrenia in this way. Okay, so that was kind of a made-up example, but showing you how you might get to the math, right? So adding up odds ratios and across multiple SNPs. Well, let's see, people have done this. So how good are we at actually predicting a trait? So we're coming back to height. And people have gone through and they've generated these polygenic risk scores and tried to see if they could estimate what people's height would be based purely off of their genetics. And remember that if you were to use an individual SNP, it would be totally worthless because even the highest effect SNPs don't tell you much. So this graph is the result of that study. I know it's probably a little hard to read, so I'm just going to try to verbally describe it to you. What you have here, you can see how there are these different kind of clusters of bars. And they're changing the number of SNPs that they're adding up the odds ratios across. So on the left, they might only be adding up the odds ratios across 10 SNPs. And then here, maybe they're doing it across 100 SNPs. And then here, they're calculating these polygenic risk scores by adding up, in this case, 10,000 SNPs. So we're no longer looking at, ApoE4, we're adding up 10,000 different SNP effects. And on the y-axis, which is really the important part, it's the amount that we're able to predict height based off just simply adding up those SNPs. And you can see that if you take the rightmost bars, that using 10,000 SNPs, you can predict about 30% of the differences in height. Since only 50% of height actually comes from SNPs, being able to get 30% is actually, that's more than half of the signal that's there, so it's actually pretty good. So a really interesting example of doing this was that this came out relatively recently, was to look at this basketball player. So this is Mr. Bradley. I don't know his first name. But apparently he's the 3rd tallest basketball player in the NBA in all history. And you can see here that he's 7'6". And so the people in the study, they said, do you think that he has a high polygenic risk score for height? Is it just that he has some weird mutation, some single mutation in a gene which is causing him to be really high? Or did he just happen to get 10,000 SNPs and every single one of those happened to predispose him to being taller? So they generated this polygenic risk curve using people from a control population, kind of matching his background. And then they calculated his polygenic risk score. And so as you probably guessed, he's this arrow that's like way outside of the curve. So in this case, it really looks like his height is due to kind of just in all those 10,000 different SNPs happening to get the one which is associated with being taller. And it all adds up to make him, quite an outlier. As a sort of second example, this is a polygenic risk score that you actually probably have access to right now if you've done 23andMe. And they have a score for calculating like what they predict your weight would be based on your genetics. And so if you've ever looked at this page, you might have noticed down here, there's this total. So this is the number of variants that they're using to predict your weight. So we are calculating height based on 10,000. Here they've decided that having 722 different variants is enough for their prediction. So that's kind of a cool little example that you may have access to already. Okay, so we have this new way of calculating somebody's risk for a disease through polygenic risk scores. So now let's talk about kind of its use in medicine and maybe its use for you. Okay, so this is a paper which came out very recently, and it's by a cardiologist, and he had a question, could we use polygenic risk scores to kind of categorize patients into high risk and low risk groups that, maybe would have different screening protocols or different treatment protocols. So the first thing he looked at was coronary artery disease. And starting over on this graph on the left, you can see that polygenic risk score is on your x-axis. So if you're on the right, you have a high polygenic risk score for CAD. If you're on the left, you have a low polygenic risk score. And these are the odds ratios versus the rest of the population. So if you're around here in this curve, meaning you have a pretty high polygenic risk score, it's like 3 times more likely that you're going to have coronary artery disease. If you move over a little bit more, 4 times more. If you move over again, 5 times more. So there's really a big difference that they're picking up on. Going to the middle, if you just compare polygenic risk scores for people with coronary artery disease versus without, just controls, you can see that on average, the polygenic risk score for people with coronary artery disease, as we expect, is much higher. And then the graph on the right, I think, is sort of the most dramatic. And so this shows you your percentile of your polygenic score. So each dot would represent probably thousands of people. At the left, these would be the people who all had like the bottom 1% of polygenic risk scores. So they're all very low. On the right, they would be in the top 1% of polygenic risk scores. And then in our y-axis is the prevalence of coronary artery disease. So you can see if you have a low polygenic risk score, you're around 1% prevalence. But then as you move to the right, you really see this sort of linear increase initially, and then just this like exponential uptick. So if you're one of these like outliers and you're in the top 1 or 2% of the polygenic risk score for this disease, then your prevalence could actually be around 10%. So that would be tenfold higher than if you were low. So they found that for coronary artery disease, and they wanted to ask, okay, what about the other common diseases? So they looked at several more. They looked at atrial fibrillation, they looked at type 2 diabetes, they looked at inflammatory bowel disease, and they looked at breast cancer. So I'll just as a quick aside, breast cancer, we had the variant BRCA1, but it turns out it's still a complex disease. And so polygenic risk scores are still very helpful for a lot of people who get breast cancer but don't have one of those variants. So in all cases, you can kind of see this trend where as you increase your polygenic risk score for a disease, you kind of have this linear increase in risk, and then it really takes off. And so I think it's a really interesting case where, it looks like sometimes it goes up 6 to 10-fold in the prevalence of this disease based on just stratifying people by this score. And so I think it's very similar to the movie Minority Report, great movie. And so in this movie, if you haven't seen it, they have the ability to kind of predict if somebody will commit a crime, but before it happens. And then the question of the movie is, well, what should we do? How do you punish them? They haven't actually committed a crime, but they eventually will. So polygenic risk score is kind of interesting. It's kind of similar in that you could do 23andMe on a baby and say, they're in this elevated profile for having coronary artery disease before that pathology has ever had the chance to develop. But you can kind of see into the future and what they might be getting. And so right now, the way people are talking about using these polygenic risk scores is largely in the areas of like disease prevention and disease screening. This image kind of shows you like the standard. It's for females, age-specific health screenings, right? And every female, these would sort of apply just across the board. But you could imagine if you had a 23andMe, if you had 23andMe data, you could go compute your risk for coronary artery disease or type 2 diabetes. And then, things like getting an annual cardiovascular exam, maybe that would be biannual if you're in the top 1% of risk for that, genetically for that disease. And so you could start to see, ways in which this could be personalized for people based on their genetic risk. Okay, so now to talk a little bit about what polygenic scores are actually available to you. If you want, if you were curious and have been genotyped, what could you actually go see? So 23andMe will do weight, which I showed earlier, but from what I've discovered on the site, I think that might be the only polygenic risk score that they make available. There's this other site called DNA Land, which is run by researchers at Columbia, I believe. And you can go upload your data there, and they will predict a couple of your traits. And so in this case, there's 8 different traits that they'll make available to you if you upload it. So yeah, if you can't read those, it's circadian rhythm, coffee, so that'd be like coffee consumption, educational attainment, intelligence, neuroticism, eye color, height, and nearsightedness. Okay, so I went through and I uploaded my data, and I wanted to sort of walk you through the results and walk you through, if you do it, how you should interpret the results. Okay, so first thing I did was height. So this says likely taller, but that was when you have males and females in. So if you look, this blue curve is actually for males. And so it predicted that I'm, 171 centimeters, so that's like 5'7", and I'm 5'9", so not terribly off. And I'm slightly, under average genetically for this height trait. Okay, you can do intelligence. So here, the x-axis is number of point gain in IQ sort of expected. And so you can see where I am, they expect that I have 1 point extra sort of in IQ versus a background population. But you'll notice it's kind of a modest effect. So the highest you can get is 5 points. Okay, and then lastly, you can do eye color. And so on the bottom, you either have blue eyes on the left or brown eyes on the right. And they predicted that I have blue eyes, which is correct. So I have like bluish, like green eyes. Okay, so those are 3 tests, things that you could go get, and now we'll talk about sort of the interpretation. So what should you believe? What should you not believe? Okay, so how much you should trust them basically goes from high on the left to low on the right, unfortunately. On the left, we have eye color, which is 100% genetic. And as I mentioned earlier, it's only 2 or 3 genes control it. So it's actually really easy, relatively speaking, to kind of predict somebody's eye color. So because of that, we'll say that 100% of the variation explained in eye color, they can basically predict it. In the middle, we have height. So height is 80% genetic, as we talked about. But in the case where you use 10,000 SNPs, you could only explain about 30% of it. They use a more limited model, so they can only explain actually 20% of the variation. So there's still a lot on the table. They could predict you to be 6'5", but the other 80% of you is 5 foot, and then okay, intelligence is an interesting trait, very studied, similar to height, and it is 80% genetic, so it's largely genetic. And when my girlfriend heard that, she went, shit, because she doesn't. given her parents. So 80% genetics. And then it turns out that the best models here are only 5% of the variation is explained. So the model that they've used on this website doesn't have very many SNPs. And even the cutting edge research, like having bigger models, they really can't explain intelligence very well based on your genetics. I'm not too sure why that is, but so going from left to right, you can't really trust the ones on the right as much. And then just to give you a better idea, variation explained, percent variation explained is a very important kind of concept for polygenic risk scores, and it tells you how much you can trust what they tell you. So here's kind of a brief example on the idea behind variation explained. So imagine here are two graphs, and we have polygenic risk score going left to right, so increasing as we go to the right. And then we are studying some traits. So let's imagine it's brown eyes and blue eyes. Here we have it as continuous sort of variable. So in this case, you can probably see that if each dot's a person, that we're pretty good at guessing what their eye color is based on their polygenic risk score. There's a clear sort of relationship. As you increase and go to the right, the Y value decreases. So in this case, you would say a lot of the variation in this trait is explained by polygenic risk score. On the right, this is probably something like what the IQ prediction would look like. So you have a slight trend, so there is some relationship as polygenic risk score increases, IQ score does go up, but you'll see that the points are just all over the place. And so you could be low for polygenic risk but have IQ. You could be high for polygenic risk and have low IQ. And so this is kind of, you'd have to take the results with a grain of salt because it's not going to be very predictive for you individually as a person. Okay, so with this, I got really interested in polygenic risk scores, and so I decided to kind of like build my own. And I'm in psych, so I wanted to make it about something brain-related. So I decided to create kind of an online tool, which you guys can all have access to as well. But it's to calculate your polygenic risk scores for different personality traits and then also some common psych diseases. So I got interested in personality because I think it's very fundamental to our everyday. It's an interesting trait, and I didn't see a tool out there to predict it based off your genetics. And I thought, people may find that interesting. So just a quick history on sort of personality. So this guy, Sir Francis Galton, founded the Lexical Hypothesis of personality. And the idea behind it was that if there's an important personality characteristic, that there will eventually be some term or there'll be some word for it in the language. And so in order to figure out personality traits, you could actually just look at language, and that would tell you what the relevant personality traits are. So they expanded, a lot of people, this is Gordon Allport, but they expanded on this idea. And so the first thing they did was, okay, if there's a word for it, then let's take every word which kind of describes personality. So he took 4,500 different words relating to personality and kind of created this distilled list. And then somebody went through and they said, well, there's a lot of synonyms here. If you're nice and friendly, how informative is that? Let's just stick with nice. And so that kind of brought it down again, and this was like in the 1940s. Then eventually, in the 1960s, somebody sort of was studying these lists of words, and Ernest Toops and Raymond Crystal, and they realized, even though we have these 171 words and now we don't have synonyms in, it turns out that certain words tend to go together. And so the idea was that you could actually just distill all of personality down to just 5 different traits. And so that's called the 5-factor model, so the big 5. And it kind of went out of style in between the 1960s and the 1980s. A lot of psychology was saying you can't predict behavior with personality, so it fell out of fashion. But it came back once it was championed by some psychologists. And so if you've heard of personality traits, you probably have heard of Myers-Briggs. 5-factor is the other one that you have run into. Okay, so here they are, just for reference. So we have openness. So if you have low openness, then you're like practical, conventional, prefer routine. On the right side of that, as you can see, if you're very open, you're curious, wide range of interests, independent. So each one is kind of an axis, you know, and you fall somewhere along that for these 5 different traits. So a big caveat here is that personality is only 5 to 10% genetic. So you'll remember that I said they GWAS'd everything, so they GWAS personality. And so because of that, if you build a polygenic risk model on personality, it likely isn't going to be very predictive of your actual personality. Most of that's environmental. But I think it's interesting to see kind of what your parents set you up for genetically and see if you overcame their influence. Okay, so I built this tool, and this is the URL for it. I also have the URL for these slides up later if you wanted to take a picture of that, which you would obviously be able to get to it. Yeah, I'll pause for a second. Okay. So, we got one more.
So we would send our data to you or?
Oh, so no. So I wouldn't have access to your genetic data at all. So it's a website, and you can upload. I built it with 23andMe data, so if you have Ancestry, I haven't supported that yet, so I'm sorry. But you basically can just upload your results, and it'll kind of spit you out these little graphs. And so, oh wait, let me just show you. So if you actually go to the site, did that? Yeah. So if you go to the site, yeah, you'll kind of be taken to this page. You just browse, upload your file. I'm not storing it. I actually am kind of new with this software, so I wouldn't even know how to store your information if I wanted to. And then once you do that, then click on, you know, you can either see your personality traits or psych risk. So click on that, and then you should see like a little loading bar in the bottom right, and it actually is calculating them like on the spot. So give it, you know, maybe 2 minutes. Yeah. Okay. So how do I? Okay. All right, so just to kind of give you a heads up with this, I'm calling it psych risk. So the other case from DNA land, they would actually kind of predict what your trait would be. To do that, I would need a lot of people's genetic data, and I'd also need to know their personality, which at this point I don't have access to. So what it's going to give you is it's going to basically cut out this case section, and it's going to give you the control population distribution, right? So it's going to place you somewhere along this distribution, and it'll say you're at the 95th percentile for this trait or this psychiatric disease. And as a quick aside, the psychiatric diseases are like autism, depression, schizophrenia, ADHD, and a few others. So it'll kind of tell you, and you know, you could sort of infer that if you're at a high percentile, then you're probably in a group which would be at higher risk, but it's definitely not diagnostic. So, okay, so this is the sort of the display that you'll get on screen, and you'll be represented by like a little sort of black colored like male sign with a gray bar going up, and they'll say your percentile. And so you'll notice that these aren't smooth curves. This is like the actual data. So this is scored relative to like 620 sort of just healthy random people with a European background. And it'll give you your percentile. So you can see, okay, I'm genetically predisposed to be, you know, maybe extroverted, low on the neuroticism scale, and so on and so forth. And for me, I went and did like a little quick check to see if it matched up with my true personality risk, which there's lots of like online Facebook tools you can use and whatnot. And in this case, it was just over 50% accurate. So that's probably the performance that we can expect. But I think that it's interesting to kind of see what genetically you're predisposed to be. Yeah. So these are some kind of like additional resources that you can use for polygenic risk scores. I showed you DNA Land. Impute.me is a really cool site that I recommend you check out. What's interesting about this site is that you can upload your data and they'll actually give you your imputed genotype, if you know what that is. It's where when you get 23andMe data, they actually only measure about 800,000 SNPs, but you can impute out or kind of guess what the rest, in this case around 8 million are, and they'll tell you that. And they have a similar tool that I had, but for a bunch of different diseases and everything. Again, that's the URL if you wanted to check out your personality or psych risk. And here's a link to these slides in case you ever wanted to reference it. And that's my contact info. So thank you all very much for your attention.
So we have some time for questions, but if you'd like to answer or ask a question, you need to ask for the microphone. Anybody have questions?
Now, we've been looking at SNPs and so on, and the actual thing now, now I've lost my vocabulary, I guess, is that you can tell me what the thing that we've seen that mediates whether things are, there's another term, something genetics, that sometimes prevents things from being expressed. Epigenetics, I guess. You know, that sort of seems mixed in here somehow too, and then of course for things like height, as you sort of alluded to, you've got environmental things that can very much. I know that they've looked at over time, or actually in some things where if nutrition is limited, then suddenly you see a shrinkage as well. So it seems like there are some things that are really mixed up, far from just genetic.
Yeah, that's definitely true. So like epigenetics, for those of you who haven't heard the term, would be, I guess it's kind of like extra genetic or above genetics. So part of what defines a trait would be, the sequence and the variation within a sequence. But there's also things which might control the expression of a gene, which aren't actually encoded in your genome, and they're just kind of like proteins, essentially, that we talked about residing on the DNA, which can control the action. So, yeah, that's definitely true. In this case, with polygenic risk scores, it's kind of just, if you take an average over a lot of things, it'll sort of balance out. But epigenetic effects certainly have an influence. So you might have, for instance, a damaging variant, which is altering some gene, and so if it was able to be expressed, then it would lead to a disease. But you might have epigenetic sort of suppression of that damaging variant, and now the variant doesn't have an effect. So, yeah.
So the other part of his question was about the environment, and so part of what you talked about was the genetic component.
Yeah.
And then a number of times you mentioned there's an environmental component as well. So can you elaborate on that?
Yeah. Well, as a geneticist, I tend to ignore the environment because it's like the other part, not my job. But it definitely plays a role, and in particular on your trait of interest. So personality, if you're a geneticist in personality, you're kind of in the minority there. Maybe you should be studying the environment. But the environment can certainly have interactions with your genotype. So what would be a good example of that?
Well, the one he mentioned was height, and there's a classic
Oh,
image
yeah.
of height of, I think it was like early, like 1914, of students at UConn, what is now UConn, University of Connecticut, and they're sort of lined up in height. I think that picture was taken in 1914. There's a redo of that picture in like 1984 or something, and the height average was quite a bit larger, simply because diet, nutrition had changed a lot over that time. It's not like the genetics of those students had changed over those 80 years between when those kids had grown up. But diet had changed a lot between kids that were born in 1900 versus a kid born in 1970 or something.
Another example was the height of men in World War I was much lower than in World War II. Same basic ancestry, but the things like nutrition and health and so on changed.
So what's your goal in your Shiny app, Psych Risk? I mean, do you see it refining it, finessing it so what is your goal?
Yeah, good question. So, I mean, my initial goal was just to kind of get my hands dirty with this Shiny app. There's already some Shiny apps which will do kind of similar things, and they've really like built them up, and it takes a lot of hours, and you also have to get it like hosted and stuff. So I'm not sure, actually. In this case, it was kind of a learning experience to see how first to compute polygenic risk scores, so I knew what was going on under the hood and could give an informed talk. And also having experience with putting something up online that was accessible. So educational first and foremost, but I think eventually it would be interesting to kind of expand it into something where it's actually predictive. So like DNA Land, if you go there, they want you to tell them, like if you want their height prediction, you have to actually tell them what your height is. And so from that, they can build a model and start predicting what people's height will be based on polygenic risk. And so in my case, if I wanted to do that, I could have people take like a personality quiz and tell me what their actual score was for openness, and then try to see how accurate of a predictive model you could get. And that could be interesting. It probably wouldn't get too predictive since it's not very genetic, but yeah.
I was just kind of curious on that impute me. What factors went into that to be able to do the calculation with more raw data?
Yeah, so are you interested in like I mentioned the imputed genotypes, but then they also have polygenic risk scores for like a lot of different traits. Or just like in general?
In general, I came from another meeting, so I apologize if
Oh,
I
no worries.
missed part of it.
So they have, so the first thing, I guess, which is really a nice service if you do have kind of your genotype, is that you can upload it, and they will take, your 800,000 SNPs and feed it through their software to guess what your genotype is at an additional 8 million SNPs or so. And so to do that, they use kind of population data where they say, okay, everybody who has an A at this site also has a T at this site. And so from that, if you only sequence one or the other, you can kind of make a best guess as to what your variant you would have at the other site. Yeah. And then for their their phenotype or their traits, they went and basically collected hundreds of different genome-wide association studies on, there's like every disease you could imagine on there. And so they kind of do a similar thing to me with my personality risk, where they assess, your percentile for a disease in a healthy population. Yeah.
I was most interested at the outset when you mentioned the work you're doing for your doctorate, and I'm sorry if I've got the terminology wrong, was on genome studies of individuals in the autistic spectrum. Being a parent of an autistic son, obviously that piqued my curiosity immediately. I guess my initial thought is the spectrum is so broad, and there are so many areas of impact, both in number and in severity. What sort of challenges have you run into at this stage in terms of the work you've done so far, without giving away the punchline of your thesis?
Yeah. Oh, I'm not hiding anything, so it's all right. Yeah, so I really what you brought up is the big challenge in autism genetics, which is that we tend to study autism spectrum disorder as a single disease, and that leads to a lot of sort of statistical problems, and it becomes hard to discover new things. And so my work, which I'll be kind of carrying out as I move forward, is to try to break groups of patients with autism or individuals with autism into different kind of clusters based on the medical phenotypes that they have. So maybe you have autism with intellectual disability and language disability. We'll study that group of people as like a single disorder. Or if you have GI sort of digestion issues and autism spectrum disorder, then that could be a group. There are disadvantages to that approach, but it's kind of the way things are going in autism genetics, is to try to parse the spectrum genetically, and so that's what we're currently working on.
Well, our time has run out, so we'll go ahead and take this opportunity to thank Leo for his presentation. And I hope to see you all back in October.